Hello, everybody. Today we're going to be talking about flow cytometry. So, what is flow cytometry? Well, flow cytometry is used to count and analyze the size, shape, and properties of individual cells within a heterogeneous population of cells. Flow cytometry data is quantitative and it can be thoroughly analyzed by specified FC software programs. So once a sample of individual cells, tissues, or organisms has been prepared, it's placed into the flow chamber of the flow cytometer and it's constricted into a line of cells within a fluid medium, which is then diverted to the path of the laser. So some of the basic principles is that it's based on the measurement of light scattered by the samples and the fluorescence observed when these particles are hit by the laser beam. A sample of cells will be inserted into a flow chamber, which then suspends them in a sheath fluid medium to be moved through the instrument. A laser beam is passed through the medium as the sample flows through the instrument. The laser beam focuses on a single individual cell at one time. So by analyzing the scattered and fluorescent light emitted by the sample cells they pass through the beam, the unique properties of the cells are able to be seen. So these are some of the basic components and instrument parts of a flow cytometer. We have a laser light source. We have a fluidic system that transports the sample through the instrument. We have the optical system, which diverts the light into the detectors. We have the detectors, which here is 3 fluorescence detectors labeled FL1, 2, and 3. And then a side scattered light detector and a forward scattered light detector. And we also have the electronic system, which importantly is analog data to digital data conversion that then goes to the computer to be processed by the software. Here's a little bit of an expanded view. We have the laser, fluidics, optics, detectors, and electronic system. So the first part of the instrument is the fluidic system. And the purpose of the fluidic system is to move the particles in the fluid system into the path of our laser beam. The sample will be injected into a sheath fluid inside of the flow chamber. This sample is then focused by injecting the sample into the center of the sheath liquid stream. You can see they're up in a line. So this fluid stream moves the sample and restricts them to the center of the stream. This is really important. So the detectors can detect any movement or any break in the laser, and that's how we know the information about the single cells. And this is the next part, the optic system. So after the sample passes through the laser, the light emitted and the fluorescent signals are then diverted to a photomultiplier tube or a photodiode that will collect the signal. To get the wanted specificity of a detector for a particular fluorescent dye, filters are placed in front of the tubes. And you can see some of these optic filters here. These filters ensure that only a narrow range of wavelengths will reach the detector. I have our detectors here. So with those two parts, we move on to the electronic system and the side scattering and forward scattering detectors.
So continuing with the optic system, we're going to be talking about two different kinds of detectors. The first detector will be the FSC, forward scattered light, and then the side scattered light, the SSC. So remember, light scattering results from particle deflection, and this is going to depend on the physical properties of the particles being inserted into the machine. The forward scattered light detector, FSC, it is proportional to the cell surface area or size of the cell. It is a measurement of diffracted light. Side scattered light detectors, SSC, indicates cell granularity or internal complexity of the cells being put into the machine. SSC is a measurement of mostly refracted and reflected light, so that's how it's going to differ from the FSC. And then the diagram to the left is a very simplified version of these detectors. Essentially, the sample is going to be inserted into the collection vessel, and then a laser beam is going to be passing through the sample, and then it's going to be transmitted to the detectors, which will eventually be transmitted to the computer software of choice. And then this diagram here is much more in detail than the diagram we previously looked at down here. Essentially, we're going to have the sample be inserted into the collection vessel, and then, as mentioned before, a laser will be passing through the sample, and then that laser will eventually make it all the way to one of these photomultiplier tubes. And then there are a series of dichroic mirrors depending on the instrument setup, and then eventually it's going to be passing through an electronic detection system and then to the software of choice. And then this is a little bit more in detail on the fluidic system, but we're going to cover that in a different section of the presentation. So since we spent some time on the fluidic and the optical systems, we're going to be talking a little bit about the electronic system. So once the sample is actually inserted into the collection vessel and the laser is going to be passing through the sample, the light signal is going to strike a photodiode or photomultiplier tube, and this is going to multiply the relative number of electrons into a stronger electrical current. And the type of information conversion is going to be analog to digital. Essentially, the electrical current is going to go through an amplifier, which is going to be converted to a voltage pulse. And the max height of the pulse is achieved when the maximum amount of scattered or fluorescence is achieved. There are several applications of flow cytometry, specifically in biomedical sciences. It does work very well with understanding fluctuations in immune cell populations as indicators of overall health. So healthy donors donate tissue samples, flow cytometry is performed, and then data acquisition eventually results in a computational analysis. This computational analysis is typically paired with coding applications. These coding applications produce powerful statistical models. This is an example of a statistical model, and essentially this allows researchers to understand certain impacts on immune cell populations in healthy and unhealthy individuals. There are several advantages to this technique. For one, it does allow for high throughput single cell analysis. It does see a lot of usage in marine and mammalian research. If researchers are concerned with cell population fluctuations in DNA and RNA, they're going to be using this kind of technique typically. It does help with understanding conditions of desired cell populations in both virtual and in vivo settings. It does have potential to be a stepping stone for personalized science and medicine, but more research is needed. Like any laboratory technique, there are certain disadvantages of flow cytometry. For one, it is a very slow process. So older flow cytometry models can really only do about 106 cells per hour, and they do produce incredibly large data sets, as shown down here. These incredibly large data sets do take a long time to analyze. Hopefully, as time goes on, the technology will progress and the speed problem will be circumnavigated. Really, to analyze these data sets, there must be incredibly efficient workflows in place and statistical processing models to actually interpret these large data sets. So we wanted to give an example of a flow cytometer. Specifically, this is a flow cytometer and a self-sorter, so this is a little bit more of an advanced machine. And this machine is the BD FACS Area 2. Moving forward, everything here is going to be extra information and will not be on the quiz. So traditional flow cytometers are really going to consist of three systems, and we've really already talked about these systems earlier in the presentation. They're going to be composed of a fluidic system, electronic system, and an optic system. So remember, the fluidic system is going to be composed of a buffered saline solution, and it's pressurized to deliver and concentrate the sample to the laser detector. The optical system is composed of lasers, photomultiplier tubes, and photodiodes. Each serve to generate visible and fluorescent light. Each signal is used to analyze the sample. So the fluorescent light is actually going to be directed via dichroic filters to the detectors and bandpass filters. This is going to serve to choose specific wavelengths of light needed to analyze the sample. For most machines, we were able to find the common wavelengths are typically about 355 nanometers, the UV region, to 640 nanometers, red. More laser wavelengths are available depending on certain applications. So continuing with extra information about traditional flow cytometers, the electronic system is going to serve to convert the data from the detector to later be interpreted by the computer and the software. For the Area 2, this is the supporting software that is most current. And even though we're talking about some extra information, the basics still is the only thing that you're going to be needing to know for the quiz. So for flow cytometry, essentially there's going to be a photomultiplier detector. There's typically going to be two photomultiplier tubes. Those are going to be serving as light detectors, obviously. There's going to be one at 180 degrees and one at 90 degrees to the laser. And these are going to be used to measure both forward and right angle scattered light. So continuing with extra information, specifically the machine that we chose as an example, the BD-FACS Area 2, it's also known as a cell sorter. So it is a flow cytometer, but it's also a cell sorter. Cell sorters are unique in the way that they can actually purify and collect samples for later analysis. So the operator can choose a certain population of cells and particles to be deposited into a collection vessel, which can be interpreted later on. The cells and particles are chosen either positive or negative for the desired parameters. This sample will be separated by oscillating liquid sample streams at high frequencies. Drops are then generated, which are either positive or negative, and will be passed through metal deflection plates and then are directed to specified collection vessels. And the collection vessels can either be tubes, slides, or plates. So we're going to be ending our presentation with a little bit more about our chosen machine, which is the BD-FACS Area 2 cell sorter. We're going to be going over a little bit about the instrument-specific components, specifically the excitation optics and a couple of the different detectors on this machine. Even though this is extra information, it is very similar to the detectors that we talked about earlier in the presentation. So specifically the excitation optics, they are class 3B lasers that are fixed upon coarse cuvette flow cells. The coarse cuvette is gel coupled by refractive index matching, and this is going to be ensuring sample collection efficiency. So the two different kinds of detectors are a little bit unique. There's a forward scattering detector, which is composed of a photodiode with a 48810 bandpass filter, and then a side scattering detector, which is actually a photomultiplier tube with a 48810 bandpass filter. And that is the end of our presentation. Thank you very much.